Thanks, Steve, for the invitation, and thank you for coming over to the lecture. When I started to prepare the lecture, I just feel how much work actually I have done in the past 10 years since I left UCLA. But again, I feel you guys are so lucky, whether you just started your career or you're already established. So the short network course will benefit everyone, including myself. So today I'm going to talk about the multiscale network analysis. So in the last few days, actually, you already heard a lot about WGCNA. That is a very important network approach, and it gives you a lot of basic concept of how the genes organize into high-level patterns like cluster or module. So today I'm going to talk about how to use this method and also use this method with other types of networks to identify novel pathways and novel and driver genes. I'm from Mount Sinai. So if you have no idea about Mount Sinai, this is where we are. So it's right in the middle of Manhattan, and so it's one of the top 20 medical schools, and but we have very small small body of graduate students. Okay, so this work actually won't be possible without significant guidance and input from Professor Zhang, who is here today, and also Dr. Eric Schott, who is the chair of the genetic department and also the director of the Icahn Institute of Genomics and Multiscale Biology. This institute actually recently got a donation about $150 million to do this kind of systems biology approach. So back to, it's about like 8 years ago, actually, we actually developed three different types of network inference methods all simultaneously. So Eric actually published the first linear regression-based causality test in Nature Genetics 2005, and Zhang actually published a paper 2004 about how to use the genetics to infer the use genetics as a prize for basic network construction. And at the same time, Steve and I also developed this kind of WGCNA, as you have learned a lot in the last few days. So basically, those three methods developed relatively independently. And when I joined Eric and Zhang's group at Rosetta, I got a good opportunity to get exposed to all these different types of methods. So that is a very good chance for me to figure out how to better integrate those different types of networks. So basically, each type of network has its own advantages, but also its own disadvantages. For example, the causality network it deals only with pairwise and components like gene and trait, and the co-expansion network does not tell you the causal direction. But the basic network is more powerful, and it potentially can integrate all types of information under the same framework. But still, the basic network is constrained by the computation intensity, and so it does not, for example, does not allow the feedback loop in the network construction, and often the network is also quite complex, and it's not easy to digest, and just using this causal network. So there must be means to further understanding those networks based on certain integration methods. So one type of integration which we will go through all the 4 talks I will give today, is the key driver analysis. So we use this particular approach to integrate causal network and those interaction or correlation-based network. Yeah, so this is just a brief review of the WGCNA. So basically, you can define similarity between any two gene expression profiles, and then you go through 2 steps of transformation, so that you can suppress those noisy correlations and enhance the true signal. And then we use a dynamic cut-tree algorithm to define the clusters of modules. Yeah, so in the last actually 8 years since we published a paper, Peter and Steve have made a significant progress actually for this actually method to be easily used for people in the community, but there are still actually significant limitations for the method. For example, even like 8 years, we already realized actually how to visualize the network. How could you actually define those significant links? So this problem actually dragged us back to the original those unweighted approaches. So it is even actually clear now actually how we claim the advantage for WGCNA, but it is a very convenient first stop, and yet to give a quick glance of the data in terms of high-level organization, like modules. Yeah, this is just a short introduction of the causal gene relationship. As June, we will talk a lot actually tomorrow about this. So I just have 1 slide to give a short introduction. So basically, if you just look at this is the 2 causal network, and when you look at the correlation, especially in the framework of WGCNA, so you see those mutual correlations. So you basically cannot differentiate. You cannot recover this 2 on the causal chain. But if you actually make use additional information like EQT or eSNP, because we know those genetic information actually control a lot of downstream like gene expressions. And in this way, then you can actually pick up those 2 models based on the price, like EQT. And in the 2008 Nature Genetics paper, and so Zhang and shown that if you integrate like a gene expression and genotype, transcription factor, and protein interaction data, so basically you will get most predictive causal Bayesian network. So this is a short introduction for causal network. Now, but there are many other ways to construct the causal network, like Alias presented yesterday. So that is an extension of Eric's linear regression-based causality test. So that kind of causality network can be also actually integrated with other type of network, like the co-expression network. So we actually developed a formal integration method called like a key driver analysis to integrate networks, but it is in a more general form. So it's not necessarily actually they input not necessarily from the gene co-expression networks, but it can be any gene set. Like from many actually experiments, you have case control a small number of samples. You won't be able to build up co-expression network because of the small number of samples. So in that case, you can only actually define those differential expression gene list. But if you have a causal network built up based on a relevant population, and then we could actually potentially integrate those gene list and the causal network, so that in the end you can identify those key regulators among those differential expressed genes, instead of you have a list of just a list of genes from the differential gene expression analysis. Now you have a rank order, rank order the gene list, so that you can actually carry out a further experimental validation. Okay, so I will have in the next lecture, I will give more details about the key driver analysis, but here I just give you some concept. So basically, it is it's a dynamic search of the causal network to see actually which nodes in the causal network actually are more enriched for the input gene list in the neighborhood of a given node. So basically, you just search all the nodes and look at the downstream or the immediate neighborhood of this particular gene, and to see how those input nodes and highlighted here as blue nodes, whether they are enriched in the neighborhood. So, and you find out that the best neighborhood actually most enriched for the input gene list. And if this kind of enrichment is significant, then we can claim that this is a potential driver gene. And then we can actually rank order all those candidate driver genes based on some statistics like an enrichment p-value, something like that. So that's the basic concept. Yeah, if you have any question, you can interrupt me anytime. Okay. Okay. Yeah, defined network, as I explained before. So basically, you can either use Jones Bayesian network or use Elias cosine network or Eric Schwartz causal network. So basically, it is open to any network, and even you can also use the protein interaction network. So basically, we don't restrict this network here. So it could be any network and relevant, but yeah, but make sure actually it's relevant to the gene set of your interest. For example, if we are studying like obesity, diabetes genes, probably you want to use those adipose or liver Bayesian network to figure out which genes are key regulators, something like that. Yeah.
Then the arrows here represent the causality, just a kind of association.
Yeah, this is a causal relationship, and but in many cases, actually, if the causal relationship is not available and you just have the association, we found out actually this method also works. Yeah. Okay, so this is a glance of so when we talk about multiscale network, so what do they look like? So this is the typical approach. So basically, we construct a co-expression network and the Bayesian network independently. So again, like when you look at this Bayesian network, it is still quite dense and complex. You cannot immediately spot which genes are the drivers, and also it's not clear how the global structure give you a meaningful subnetwork relevant to the clinical trace of our interest. So, so one approach to further actually understand the complexity of the Bayesian network is to use the co-expression network modules to dissect this complex and hairy ball, and so that we can get a clearer visualization of the subnetwork actually corresponding to particular gene modules derived from the like WGCN or other co-expression network method. So that's the basic approach. So you have the modules here actually, and from this WGCN heat map, and then you color the Bayesian network, so you can see actually the modules actually correspond well to the different components of the Bayesian network, and eventually when you use the key driver analysis, then you will be able to derive a module-based Bayesian network so that you can easily define those driver genes. So this is how we claim the multiscale networks and the multiscale network construction and analysis. And those drivers actually will go through functional validation either actually experimentally or in silico. So we have a lot of studies to show that those drivers through this multiscale network analysis actually tend to be much more biologically meaningful than those non-driver genes. So we will repeatedly and see this kind of.
I'm not quite clear how you go from the modules to the local subnetworks. I was thinking it was just those that were the most connected, but then you brought in the QTL analysis. So can you say something more about how you get to the local subnetworks, and then what nominates the key driver?
Oh yes. So basically, it is it actually goes through this particular procedure. So you input that those blue nodes are the modules, okay, and then you look at the Bayesian network to see actually which nodes in the Bayesian network controls like a majority of the input nodes, and then you can derive such a simplified module-based subnetwork.
So that's not necessarily the gene with the highest connectivity, or
no. It's completely different because those Bayesian network actually are constructed based on different theories. When you see those hub nodes, probably most often are not necessarily the driver genes. Yeah, just because those correlations are actually very noisy, and we don't consider a lot of other information like genetic perturbations. But if you consider those information and formally integrate in the WGCNA, it could actually allow you to define potential drivers. But eventually you will go through like those Bayesian network framework or causality, that use the genetic perturbation as a variable for network construction. So just this is a short review of those work, but I will give more details about some of the approaches. So like in 2006, actually, these are the two high-profile applications of the WGCNA. So one application in Astro and another one in brain cancer. So both papers have been cited for maybe 200 or 300 times. And then 2008, actually, we published two companion papers in Nature. So the basic idea is to use the co-expression network first to identify the modules highly most correlated with the obesity diabetes traits, like BMI. And then 2008, June started to formally integrate multiple types of information for Bayesian network construction. And after that, then we actually formally integrate Bayesian and co-expression network and identify a lot of new pathways and key genes in human liver and in those inflammatory those signature, which is common to many chronic inflammation disease. And okay, so let's look at the first application. So for the WGCNA, actually, this is a pretty good success and very encouraging. So basically, we have this kind of adipose data from about like 1,000 human samples, and so we have the gene splicing, genotype, and clinical information. So we have the males and the females. So so the first step, you construct male and female co-expression network separately, and then look at how the modules actually conserved across and two gender-specific networks. So basically, we found out that this immune response and the module is the most conserved and also most correlated to the obesity diabetes traits BMI. So this is so you can see only a few modules identified at that time because we were limited by the memory, but now it is not a problem. And like at Mount Sinai, we have machine with 2 TB memory, and now I can use another machine in the Oak National laboratory. Actually, they have a machine with 4 TB memory. So basically, we can put together like about like 20 or 200 or 300,000 elements in the network construction, which can also be completed in a reasonably short time. Yeah. So this actually, and then this module goes through the causality test and the Bayesian network and to identify top driver genes for obesity diabetes. And there's another follow-up paper published in Nature Genetics actually. That is a lot of faculty at UCLA, and Dr. Zhang's work actually, she found out that about like 80% of those potential driver genes actually have a significant impact on the mouse weight when they perturbed. Okay, so the second example is integration of co-expression network and Bayesian network in human liver. So this is a cohort actually including 430 human liver samples. So again, like we have a complete clinical and genotype and gene expression information. And so these 2 types of networks actually allow us to define the critical subnetwork actually for CYP 450 system. So this subnetwork actually is particularly useful for understanding drug metabolism, because a lot of CYP genes actually involved in this subnetwork, and probably it's the first time actually we could claim like which CYP genes are the top drivers in the whole system. So this is pretty actually exciting work. And more recent work actually is about the integration of a signature. Okay, so this is a kind of integration of a signature and a Bayesian network. So the signature and the networks actually were identified actually completely independently. So for the inflammatome signature, we have actually 11 mouse models covering 8 actually different diseases, including like cancer, obesity, diabetes, and COPD, asthma. So for each model, we have both case and control. And so in this way, we can define signatures actually across all those different mouse models for the chronic inflammation diseases. And so we eventually we identify about like 2,500 inflammatory gene signature from those 12 and the mouse model. So a lot of surprise actually. Those 2,500 inflammatory genes actually conserved in many co-expression networks involved in like obesity, diabetes, and even cancer. So basically, you can see huge enrichment in those networks, the co-expression networks. Yeah, so we say like at Rosetta, actually, we constructed probably hundreds of those networks, and then we can systematically actually compare how those modules are conserved across different systems and different diseases, and how those modules actually relate to clinical traits. So this actually is a good assurance of the quality of the signature. And then we actually made use of the Bayesian network. That is a collection of about 50 or 60 Bayesian networks, and from many different tissues, and in both mouse and human. And then so here I just shown like two consensus Bayesian networks. So here you can see actually from those liver and adipose consensus networks, we use the Bayesian network, use the key driver analysis to further identify those potential driver genes for the 2500 inflammatome gene signature. So you can see actually many of even we have two different tissues, actually you can see a lot of driver genes actually conserved, like TYROBP, CD53, and even TREM2. So last year when we published a paper, actually this is actually a very much conserved inflammatome subnetwork and conserved drivers. So we published this paper in July last year, and after 5 months, and then there are two New England Journal of Medicine papers actually claim actually TREM2 is Alzheimer, later onset Alzheimer disease susceptibility gene based on the GWAS study. And in another 4 months, and we published another paper in Cell, and we identified TYROBP actually as a top driver of the inflammation response module, which actually is the top causal subnetwork we identified for the Alzheimer disease. So basically, this finding based on completely and different systems and different diseases actually tell us more about the drivers and the subnetwork for other diseases. So that is really actually amazing to also showing the power of the network analysis. Yeah. So basically, we see this kind of common inflammation drivers and the subnetworks across species and across multiple, actually many diseases. So that is pretty actually exciting. So this basically paves the way for personalized medicine. When we talk about personalized medicine, you don't have that many drugs, okay, for everyone. You have like 6 billion people, we don't have that many drugs. We have only a few thousand drugs. So basically, I think this kind of common mechanism for disease is much more important than those personalized information, is my opinion. But if you can find out that there's causal mechanisms for 60 or 70 patients, you just need a single drug to treat them. And for the rest 30 or 40% of patients, you can use individualized medicine. That's the story. Okay. Okay, so when we found out the drivers for those inflammatome gene signature, so we want to see whether those drivers actually make sense or not. Especially, we want to compare the importance of those drivers with those long driver genes. So we look at the MGI and the JAX lab, and then they have some phenotype database, so they perturb certain genes and to see whether there are some phenotype changes. So from this database, actually, we found out that in general, they, about like 10 to 15% of genes have a certain phenotype when they were perturbed. So when we look at our drivers, especially the top 55 drivers of the inflammatome signature, actually, we found out that 74% of those drivers have relevant phenotypes when they're perturbed. And so when you look at those long driver genes, so only like 40% of them have relevant phenotypes. So this actually just shows the importance of the driver genes identified through this kind of multiscale network analysis. So we also look at, okay.
The non-drivers, are those still genes that were in the subnetwork?
Yes. Yeah. Yeah, then we actually collect more actually signatures from the literature. That's almost all inflammatory-related signatures. Again, like we see those drivers are much more significantly enriched for those individual experiments derived signatures. So again, like we shown the importance of the driver genes through the key driver analysis, and just also showing the power of this kind of the Bayesian causal network. Yeah. So another application is the cancer gene network, and so we try to actually first, again, like because the input of the key driver is about like a gene list and a Bayesian network. So in this approach, we try to use different ways to identify relevant genes first. So we try to infer the genomic alteration, like amplification and deleted regions in the human genome, which is associated with outcomes of cancer patients, those good outcome and bad outcome. So we actually use this kind of wavelet transform. Again, like just like WGCNA, when I started to work with Steve on that, we bought the concept from the engineering. So that is, we use the sigmoid function and also the power function to suppress the noisy. So again, like a wavelet transform is another actually very popular technique in engineering. So used for signal processing and image compression. So this is basically is another good application of those techniques in the engineering field. Okay, so this is a basic concept of the inference of recurrent and CMV regions. So basically, you can for a cohort, you can divide the patients into two groups: the bad outcome and the good outcome, the survival long and the survival short. And then we smooth this gene suppression profile. So you basically then you compare the two group of the samples for each gene, and then you derive the T statistic. And then this then you use the wavelet transform to smooth and this kind of T statistic profiling, and then we identify those regions actually amplified or deleted. So you can see here actually certain regions actually conserved across 4 different studies. So the goal actually for us is to identify those regions actually amplified or deleted regions across 4 different studies of breast cancer. So yeah, this work actually was carried out by my former postdoc Lin Chen, who is now a postdoc at UCLA. So basically, Lin actually found out this kind of expression-based CNV regions actually corresponding very well to the CNV data. So eventually, we identified 100 online recurrent regions of breast cancer from 4 different studies. So those regions, they just give you some association, just like Inflammatome gene signature. It just tell you those genes actually are important for the model, but it does not tell you the importance, the relative importance of the genes. So we, again, we made use of the Bayesian network constructed by Zhang, and we use the key driver analysis to further identify and rank order the genes in the those recurrent CNV regions. So those larger nodes actually are the key drivers of breast cancer, and those you see those green nodes actually are the nodes we know they have a significant impact on cancer cell proliferation when we're locked down. So again, we try to assess the importance of those key driver genes. So at Merck, actually, we systematically locked down 3,000 genes in 4 different breast cancer cell lines, and we identified about 200 genes actually consistently impact the proliferation of cancer cells. And then we overlay those driver genes, non-driver genes, with this siRNA hit signature. And then we found out those drivers actually, especially the global drivers, are 8 times more likely to impact the cancer cell proliferation than those recurrent, then the genes on the non-recurrent regions. Yeah. So again, those the regions we identify actually are also very important because it is about 2 times more likely to impact the cancer cell proliferation. So again, this, you can see this trend actually, from the non-driver genes to the local driver to the global driver. So the trend is very clear that those driver genes have much more biological importance. Yeah. Okay, so I still have 20 minutes, and so I have another 2 types. So basically now we talk about the multiscale network construction and key driver analysis, and those multiscale network actually can also be used for prediction and for classification. So this is actually one experiment that we did at Rosetta at that time actually. So Gaurav Pandey was an intern at my group, and he's now an assistant professor at Mount Sinai. So this work actually was identified as one of the breakthroughs in the field of computational biology in 2010 by Nature Biotechnology. So the problem we actually try to solve is to predict those synthetic seq lethal interactions, because this kind of lethality interactions involves a huge number of combinations. So it's not feasible to actually test every pair of genes for this kind of interactions. So prediction actually is very important. So this is just a very overall view of the known synthetic lethal network. So only a small portion of the in yeast, this is in yeast, actually only a small portion of the pairs actually were tested at that time. So we actually made use a lot of network features, like for the co-expression network, we look at the co-module membership. We also look at the topological overlap between any two genes and the correlations. And also we use the Bayesian network and protein interaction network. So we use, and then we actually use this similar to topological overlap, actually we use for WGCNA. So we extend this concept to multiple networks. So you can see actually interaction between those two genes, not only just from one single network, so we also consider additional network. So this actually is a more kind of formal integration of like multiple types of networks, and then allow us actually define more features for classification. So you can see actually a lot of features have good actually power to differentiate those pairs with synthetic lethal interactions and the pairs without interactions. Yeah, especially when you look at the overlay features between the PPI and the semantic similarity, so you can you have a good separation for the two types of samples. Okay. So we actually use a very sophisticated method. It's like a combine. We use 6 different classifiers, and then use this kind of logistic AND method to integrate those methods. And you can see here actually this integration of multiple classifiers is the best performance, and also it is much better than the best method at that time, actually 2010. So we actually, based on the prediction, the predicted synthetic lethal interactions, we go through the interactions between transcription factors, and so this is probably is the first transcription factor-based synthetic lethal interaction network in yeast we identified. So this we can see it. So here I just shown the interactions between two families of genes. One is a YAP, another one HYPE. So in literature, we found out, and there was actually a double lockout experiment for YAP1 and YAP2. Like many years ago, and so they shown this kind of synthetic sick lethal interactions between these two genes. And also in literature, HYPE2, 3, 4, 5 actually they form a complex. So, but here actually we use this kind of prediction method to show that there are a number of synthetic lethal interactions and between the genes in this complex. So basically, this kind of prediction actually supported by, in some way, by the literature. But for validation of those networks, just as the drivers from the previous approach, actually requires enormous method. So that's also the limitation for the field. So we don't have that many validation data set, but we have a lot of hypotheses generated. Okay, so the last example is the multiscale networks in Alzheimer's disease, and we'll give more details in the afternoon on the talk. So basically, so in this study, we have both control and control normal samples and samples, brain samples from Alzheimer's disease patients. And so again, like the number of samples is huge. So we have over 370 samples from the Alzheimer's disease patients, and also have about like 150 samples from normal control. So we actually build up this kind of multiple tissue co-expression network. So it's different from the traditional single tissue-based co-expression network. So we concatenate all the genes from multiple the tissues, so that like for each tissue you have 30 or 40,000 genes. Then when you concatenate all those genes, then you have 120,000 genes, and that's how we build up the network. And because we are not only interested in the single interactions between genes in single tissue, but also we try to capture the interactions across multiple tissues. And because in this study we have tissue samples from 3 different brain regions, and then we actually quantify the changes of WGCNA derived modules. So in this case, you can see actually this is 40,000 most informative genes from 3 different brain regions. And then for each module, and we formally define we call like a module differential collectivity, MDC, to define whether actually the interaction actually is gained or lost. So here you can see this particular module actually gain and collectivity by 91-fold. But for those actually neuron-related module actually lost the interaction by like 40%. So all this actually makes sense. And also for inflammation, we see increased interactions. So we actually further rank order. So we identify 111 gene modules. Some of them are specific to certain tissue, but many of them actually also includes genes from multiple tissues. And then we actually rank order those modules based on the correlations to about like 30 clinical traits derived for this particular cohort. And so we further actually rank order. So you can see actually 2 hotspots here in this correlation map. So this hotspot actually correspond to the immune response modules. And this actually corresponds to nerve enhancement. So in the end, actually, we rank and order those modules based on the number of traits correlated with the module, and also differential expression, also enrichment for EQTL. So it turns out, you can see here, actually, immune response module is the top ranked module in terms of relevance to AD pathology. And so we also build up a basic network for each of the module. And for this top ranked immune response module, we identify actually TYROBP as a top regulator. So we look at the phenotypes of the microglia, the mixed culture of microglia cells and neuron. So the experiment I didn't show here actually because we have another manuscript actually in preparation. So the experiment shows that the perturbation of TYROBP actually, especially the overexpression of TYROBP, increase the uptake of amyloid beta. That turns out to be good, but the consequence actually is very bad because we see actually shorten the neural length after overexpression of TYROBP. So that is the phenotype relevance of this top driver of immune response. So we also actually look at the genes responding to the perturbation of TYROBP in the cell line system. It's microglia cells. So we see actually those overexpression or lockdown signature of TYROBP actually very significantly enriched in the network we predicted, constructed from the data. So you can see a huge enrichment here. So that's all the and the different applications of multiscale networks analysis. So like in summary, so we found out actually those networks actually especially based on large-scale genetic and genomic data, are capable of painting a global landscape of interactions between the genes, and also allow us to identify particular subnetworks actually correlated with certain clinical traits of our interest. So we could also use this kind of multiscale network analysis to identify those novel pathways and the novel targets, and many of them have been validated at different levels. So we believe that now more and more large-scale data sets actually available. Like now we have GTAX and TCGA, so there are hundreds of thousands of samples there. So there is actually a great opportunity for all of us to use the network approach to further and identify new pathways and new drivers underlying those complex biological systems of complex human diseases. The work actually is impossible without support from actually many collaborators. So I particularly want to thank like Dr. Zhang and Eric Schad, and also a senior director at Merck at that time, Weiler, and also my postdoc at Sage, Chris, and G, and Trenley, and many other, and also like Steve Horvath. And like 10 years ago, I have no background about bioinformatics or genetics, but I don't know why Steve actually gave me the offer. So that's quite a miracle. I still don't understand today, and so that's so to keep this lesson. So in my group actually I pick up and people with much and diverse background. Some people without any biology background, like Wang Ming, and some people like Yongzong and Shendong actually don't have any computation skill. So I try to make another experiment here actually in my group to see whether we can make some magic here. Okay. They overlap sometimes. Yeah, like TYROBP, like HCK, those are also the hub genes in the WGCNA modules. Yeah. Yeah, so the problem is that from those interaction or correlation networks, we just cannot claim that the drivers. We know that the hubs, and then you have to do a little bit more work to further actually differentiate those kind of noisy correlation and a true correlation. So like we can use a Bayesian or alias causality test. So in the paper we published in Sleep, actually we basically use this kind of concept. So we didn't use a Bayesian network. Instead we use the causality network. So we derive actually similar performance. Yeah.
You said you use the Bayesian network for each module. You can get the causality with the Bayesian network because it's not time series or knockdown data. How can you do that?
Yeah, you can. Yeah, I will leave this question to June. So probably he can give you a better answer tomorrow. Yeah.
Peter, do you find it a problem for the causal analysis that if you concentrate on a module, then a lot of the genes are highly correlated? And if you do, how do you deal with that?
Yeah, again, like this is a very simplified visualization of the modules, and because if you really want to look at those very dense correlations, you can never figure out the drivers. Yeah. Yeah, that is also limited. Even for the Bayesian network, it is also limited by the a lot of assumptions that are not biologically meaningful.